Hello dear students, today's lecture topic: History of Immunology, Evolution of the Immune Response, Nonspecific Immunity, the Anatomy of Immune System, Antigens. Before we diving into the structure and detailed compounds of immune system, any study of immunology benefit from overview of immunology history. Let's go back in time a couple thousand years. Before we knew that the immune system was, before we knew the pathogenic microbes exist, people were still able to recognize the fundamental principles of immunity. In description of a terrible plague that swept Athens in 400 BC, the Greek historian Thucydides wrote, "For the same man was never attacked twice, never at least fatally." It wouldn't be until centuries later than discoveries of Louis Pasteur which confirmed and completed the germ theory of infection disease, as well as Robert Koch's studies of etiology of infection disease, notably tuberculosis, raised a question of fundamental importance. Is the host a helpless prey of pathogenic microbes, or it's equipped with efficient defense mechanisms to combat its invaders? You can say that immunology started the last quarter of 19th century with two major discoveries. The first of these was Elias Mechnikov, identification of phagocytic cells which engulf and destroy invading pathogens. This laid the basic of innate immunity. The second discovery was Emil Björk's and Paul Ehrlich's identification of antibodies which neutralize microbial toxins. This became the basis for acquired immunity. These findings also led to the distinction between cellular and humoral immunity. As you see, immunology didn't take off as an academic discipline until scientists began using experiments and deeper to uncover the major components of humoral and cellular immune system. As you see, we have a lot of discoveries previous centuries. Immunology is a branch of medicine and biology that covers the medical study of immune system in different organisms. So, immunity refers to the body's ability to prevent invasion of pathogens. Let's start with the main types of immunity. Individual immunity consists of non-specific or innate immunity and specific or acquired immunity. The innate immune system is the first to respond to pathogens and does not retain memory of previous disease. Innate immune response are evolutionary older than adaptive responses, and elements of innate immunity you can find in all multicellular organisms. If a pathogen persists, the adaptive immune system will engage the pathogen with specificity and memory. Active immunity results when exposure to a disease organism triggers the immune system to produce antibodies to this disease, and active immunity can be acquired through natural also immunity or vaccine induced immunity, or how we say artificial. Natural immunity is acquired from exposure to the disease organism through infection with the actual disease. Vaccine induce immunity, in our hand, is acquired through the introduction of a killed or weakened form of the disease organism through vaccination. We have another type also, passive immunity. Passive immunity is provided when a person is given antibodies to a disease rather than producing them through his or her own immune system. Why is passive? Yeah. A newborn baby acquires passive immunity from its mother through the placenta. This kind of natural passive immunity. People can also get passive immunity through antibody-contained blood products, such as some drugs like immunoglobulin, which may be given when immediate protection from a specific disease is needed. The major advantage to passive immunity is that protection is immediate, whereas active immunity takes time, usually could be several weeks to develop. However, passive immunity lasts only for a few weeks or months. Only active immunity is long-lasting. So, let's introduce some deeper information about these types of immunity. So, innate immunity is the body's first line of defense against pathogens. It is general and non-specific, which means it does not differentiate between types of pathogens. The adaptive immune response is more complex than innate. Adaptive immunity is a type of immunity that is built up as we expose to diseases or get vaccinated. And adaptive immune response is much slower than innate immunity, and also includes a memory that makes future responses against a specific antigen more efficient. The innate immune system is assumed to have arisen more than 600 million years ago. Invertebrates was the first that used three major innate defensive subsystems mechanisms: phagocytosis by blood or body cavity cells, protease cascades that lead to fluid clothing, melanin formation and opsonization, and the production of the wide variety of antimicrobial peptides. Several compounds of the adaptive immune system, including immunoglobulins, T cells, B cells, receptors, and major histocompatibility complex, are assumed to have arisen in first jaws vertebrates. More important that the adaptive immune response is unique to vertebrates. One of its core organ, like thymus, which exists in all vertebrate species. Let's check our immune system of our body. So we have and our immune system consists from primary and secondary immune organs. Primary or central lymphoid organs, including bone marrow, thymus, are sites where lymphocytes first express antigen receptor and attain phenotypic and functional maturity. We call this education. And in bone marrow and in thymus are sites of maturation of B cells and T cells respectively. Secondary or peripheral lymphoid organs include lymph nodes, spleen, and compounds of the mucosal immune system, where lymphocytes responses to foreign antigen are initiated and develop. These organs are not molecularly organized in ways that optimize the cellular interaction necessary for initiation of adaptive immune response. So, the function of primary and secondary lymphoid organs are lymphopoiesis, antigen-independent stage of differentiation, development and acquisition of immunological competence, positive-negative selection in thymus, and control of immune response. In secondary immune organs, like we said, antigen-dependent stage of differentiation. Also can be development of B cell specificity, like affinity maturation, and activation of the immune response. Let's now describe each compound of immune system in more detail for the perspective of immune activity. First up, from the bone marrow. The bone marrow is the site of generation of all cells and the site of B cell maturation, the generation of all blood cells called hematopoiesis. Red blood cells, granulocytes, monocytes, dendritic cells, mast cells, platelets, B, T lymphocytes originate from a common hematopoietic stem cell in the bone marrow. The main function of bone marrow are hematopoietic function, hematopoiesis, B cell differentiation, also the storage of plasma and memory B cells. The thymus is the site of T cell maturation. It is a bilobed organ situated in the anterior mediastinum, which involves after puberty, so this is not detectable in elderly people. And each lobe is divided in multiple lobes by fibrous septa, and each lobe consists of outer cortex and inner medulla. Before birth and through childhood, the thymus is instrumental in the production and maturation of T lymphocytes. The vast role of thymus is have immune cell distinguish between self and non-self. The answer to this question lies in the processes of positive and negative selection. Therefore, T cells know to attack and kill invading bacterial cells while leaving other neighboring cells alone and unharmed. Also, the thymus produces and secrete thymosin, a hormone necessary for T cell development and production. The spleen weighs approximately 150 grams in adults, and it's located in the left upper quadrant of the abdomen. The splenic parenchyma is divided in a red pulp, which is composed mainly of blood-filled vascular synocytes, and lymphocytes-rich white pulp. The white pulp contains the cells that mediate adaptive immune responses to blood-borne antigens. The spleen is a highly vascularized organ whose major functions are to remove aging and damaged blood cells and particles such as immune complexes and opsonized microbes from the circulation and initiate adaptive immune responses to blood-borne antigens, like we said before. The next one. There are approximately 500 lymph nodes in the human body. They encapsulate, like spleen, secondary lymphoid organs located in many parts of our body along lymphatics, and where naive B T cells respond to antigens that collected by lymph from peripheral tissues. So, extracellular fluid, this is called lymph, is constantly drained from tissues through lymphatics into lymph nodes and eventually into blood. Microbial agents occur in soluble form and within dendritic cells in the lymph to lymph nodes, where they are recognized by lymphocytes, like this one. So this means for antigen-specific T and B cells to activate, they must first suffer exposure to antigen with aid of antigen-presenting cells, like we called dendritic cells. So, some conclusion. And this organization permits the rapid delivery, the components of immune system, the peripheral and central immune organs permit the rapid delivery of innate immune cells to site of infection and permits a small number of lymphocytes specific for any antigen to locate and respond effectively to that antigen regardless of where in the body the antigen is introduced. We have a lot of function of lymphocytes, we have a lot of function of spleen, lymph nodes, thymus and bone marrow. So this is the whole of the immune system, and this system needs works precisely. We have helpers for these organs, and these organs consist from immune cells and our body, included in our blood immune and our tissues immune cells. So the cells that perform the majority of effector function of innate and adaptive immunity firstly are phagocytes, including neutrophils, macrophages, monocytes, mast cells we have, basophils, eosinophils, dendritic cells, and also T and B lymphocytes. Compounds of the innate immune system include also physical barriers such as skin or gastrointestinal tract, cellular components we have, and third one, humoral components like pattern recognition receptors and immune cells, including macrophages, neutrophils, and complement system. So let's find out some details about non-specific immunity. So, as we talked, innate immunity is non-specific, which means it does not differentiate between types of antigen. Means take an immediate effect upon encounter with anything that our immune system deemed to be an unwelcome guest. But our immune system knows when to make the immune response. The ability to recognize and respond to foreign entities is essential to the operation of the immune system. Thus, a crucial role of the immune system is determine what is foreign, or what immunologists often call non-self, from what normally present in the body in self. As a consequence, the cells and molecules that are part of innate immune system are preoccupied with detecting the presence of particle molecule patterns that are typically associated with infection agents. PAMs, pathogen-associated molecule patterns. Charlie Genevay dubbed such molecules as PAMs, and it is these structures that trigger activation of innate immune system by pattern recognition receptors, PRR. Immune cell recognize PAMs via germ-like encoded pattern recognition receptors, PRR receptors present in all immune cells. Currently, PRR families are divided into transmembrane receptors and those that reside in intracellular compartments. The former include toll-like receptors, lectin receptors, fecolines, and later the nucleotide binding oligomerization domain node-like receptors and some RIC receptors. The family of toll-like receptors can be studied most extensively. Different toll-like receptors appears to play a crucial role in the initiation of the immune responses by recognition different pumps. You see, you have different pumps by E cells compounds and microbial compounds, and that toll-likes are able to recognize a variety of pumps including lipopolyetherines, lipopeptides like toll-like receptor 2, toll-like receptor 3, peptidoglycan in gram-positive bacteria, polyanions like scavenge receptor, mannose contain carbohydrates, mannose binding protein. Also, they can activated by double-stranded RNA, toll-like receptor 3, flagellin, bacterial flagella, toll-like receptor 5, and also a lot of another receptors, because in human we have 10 types of toll-like receptors. We called of all of these types of pumps pattern associated molecular that you can find in some viruses, yeast, fungal cells, bacterial cells, all called of this. You can group all these pumps into some word, antigen. So, antigens are agents like microorganisms, particles, cells, molecules that can trigger the immune response. We can categorize antigen by origins, exogenous antigens. These antigens originate from outside and they are foreign to host body. Viruses, bacteria, most important, and some parasites and fungi. These antigens enters to the body through inhalation, injection, and they start circulating in the body fluids and they can be caught by the antigen presenting cells such as dendritic cells, some macrophages. The engulfment of those exogenous antigens by APC, antigen presenting cells, are mainly mediated by the phagocytosis, something like this. Another major group, this is endogenous antigen. These antigens originate inside our own body. These antigens are body's own cells or subfragments or compounds or the antigens products that are produced as a result of normal cell metabolism or because sometimes of viral replication in host cells. The endogenous antigens are perceived and presented also by the antigen presenting cells to CD8 cells, cytotoxic T cells, and activated immune response. Also, we have some classification of antigens by the complete antigens, or we call this immunogene molecules that induce a specific immune response by themselves, and incomplete antigens is substances which can bind with specific antibody but cannot induce immune response by itself. Another term, as you see, haptens. Immunogenicity of an antigen is determined by four properties. This is the main two properties of antigen. They classified by these properties: immunogenicity. Immunogenicity determined by four properties like foreignness, molecular size, chemical composition, heterogeneity, and most important, the ability to be processed and presented on the surface of antigen-presenting cells. Another property, antigenicity, the ability of foreign molecules to combine specifically with antibody. You will be introduced with these properties as with antigens and both with antibodies in next lecture. So, okay, this is classical bacterial antigen structure. We are presented with E. coli bacteria, classical gram-negative bacteria. The cell wall of gram-negative bacteria features its outer membrane with lipopolysaccharides. This is O antigen, or somatic or cell wall antigen. This is a highly variable surface lipopolysaccharides responsible for all gram-negative infections pathogenesis. Also, many bacteria have capsules of polysaccharides. This is K antigen. K antigen are the capsule, a coat of surface of bacteria outside the cell envelope. The capsule can protect bacteria from phagocytosis, the most important defense mechanism evolutionarily originated in bacterial cells. Also, you see that some bacteria has another variant of K antigen, this called V antigen, in case with Salmonella typhi. Attachment of pili or fimbri also facilitate adhesion to host cells. It can be involved in pathogenesis. Motile bacteria possess flagella. The last one is called H antigen or flagella antigen. It's protein antigen based on flagella structure. This is also help bacteria in pathogenesis process. Due viruses pathogenesis has a different strategy from bacterial. Viruses have absolutely different structure, and based on this, they have different antigen structure. Firstly, this could be genetic materials made from DNA or RNA. A protein coat that protects these genes, this is called capsid. In some cases, like HCV, herpes simplex viruses, first an envelope of lipids that surrounds the protein coat when they are outside the cell. This means called supercapsid or envelope. Okay. The important ability of the immune system not only to recognize but also limit microbes early during infection is based primarily on the employment of direct lysis of pathogen, opsonization followed by phagocytosis, direct phagocytosis via cell associated by different families of pattern recognition receptors, enhancement of phagocytic cell function, production of antimicrobial proteins, production and regulation by this production of immune response, differentiation of effector cells to more active states. That means, in some case, innate immunity, first step to adaptive immunity. Okay, what I want to show you that we need stop at the cellular compounds of the innate immune system, which includes phagocytic macrophages, granulocytes, cytotoxic natural killer NK cells in this case, eosinophiles, dendritic cells, and so on. As you see, you have a lot cells involved in cellular innate immune response. We can stop only with few cells, and you see that they have so many functions and they adapted, and also their line between innate immunity and next step to the adaptive immunity. Firstly, we start with my favorite immune cells, macrophages. Some macrophages are always present in most tissues and functions as guard of infection, while other phagocytes, including monocytes, neutrophils in this case, are recruited into infected tissue in response to microbes or signals generated by the guard cells. As you see, this looks like macrophages because of their body structure. Okay, let's deeper. Macrophages originate from blood monocytes that leave the circulation to differentiate in different tissues. Some examples of macrophage in specific tissue. Okay, alveolar macrophages in lung alveoli, Kupffer cells in the liver, microglia. This is resident macrophages, resident phagocytes in central nervous system. Like I said, macrophages are able to detect products of bacteria and other microorganisms using a system of recognition receptor like toll-like receptor, a lot of toll-like receptors. Also, macrophages can be a line between innate immunity and adaptive immunity, such as antigen presenting cells. All antigen presenting cells have on their surface MHC complex. The essential role of Z phagocytes, maybe not all roles, but some essential roles that phagocytes played in innate immune defense against microbes is demonstrated by the high rate of lethal bacterial and fungal infection in patients with low blood neutrophil counts caused by bone marrow cancer or chemotherapy and radiation for cancer. As you see, tissue macrophages perform several important functions in innate and adaptive immunity, such as a major function of macrophages in host defense is to inject microbes by the process of phagocytosis and then to kill the ingested microbes. This means, first, the most important phagocytosis function. Also, macrophages are activated by microbial substances to secrete severe different cytokines that act on endothelial cells lining blood vessels to enhance the recruitment of more monocytes or other leukocytes from the blood into sites of infections, like this one, yeah. And also, in addition, they play part in the cleaning up process after infection or sterile tissue injury, like macrophages have on their surface scavenge receptors, and scavenge receptors definitely have this function. Also, macrophages serve as like we said, antigen presenting cells that display fragments of protein antigens to and activate T lymphocytes from naive T lymphocytes based on which MHC complex would be activated. We polarize these T cells in Th1 or Th2 immune response, or in some cases cytotoxic CD8 cells. This function is important in the effector phase of T-cell mediated immune response. Okay, guys, we just emphasized that macrophages could recruitment of more monocytes and neutrophils from the blood into sites of infection. And another type of cells that could be also most important cells in cellular innate immunity, they are neutrophils. Neutrophils are the most abundant population of circulating white blood cells and the principal cell type in acute inflammatory response. Neutrophils nucleus is segmented in 3 to 5 connected lobules Because of their nuclear morphology, neutrophils are also called polymorphonuclear leukocytes. Some features of these immune cells that the cytoplasm contains two types of membrane-bound granules. The majority of these granules are called specific. Granules are filled with enzymes such as lysozyme, collagenase, elastase. Another type of granules called auxophilic granules contains enzyme and other microbicidal substances like myeloperoxidase, catepsine, different defensine, something like this. Because of this, neutrophils have unique killing mechanism. These three types of these mechanisms displayed on this slide. This could be degranulation and post-killing of bacterial cells, and also some netosis formation. Okay, to emphasize the unrepeatable role of these cells, immunologists called them professional phagocytes. Our neutrophil and macrophages, who are particularly adept at phagocytosing and killing microbes. Ilya Mechnikov was the first to discover phagocytosis. He was the first scientist to observe the uptake of particles by cells and realized the importance of this process for the host response to injury and infection. And nowadays, you can't imagine some innate immune defense without phagocytosis against bacterial infection. This is main compounds, main mechanism of our line of defense against bacteria. Phagocytosis can be divided into 6 main steps. You look at these steps on this slide. This is a first one here. This is recognition of the target particles. After recognition of the target particles and membrane activation, phagocytosis receptor initiate like signaling cascades that remodel lipids in the cell membrane and regulate the actin cytoskeleton in order to extend the cell membrane around the particles. A next step would be phagosome formation and also phagosome maturation. Phagosome maturation, that means phagosome merge with lysosomes, with enzymes in phagocyte cells. Next step, most important, the main step, killing and digestion, and also a release of degradation products. This means that phagocytosis steps start with inactivated macrophages or inactivated neutrophils, and they always end with zero activated also state. That means that we need activate phagocyte cells. Phagocyte cells need done their work, and after scavenge, for example, scavenge receptor of macrophages could help these cells return to zero active status. Pathogen killing can occur in two ways. The oxygen-dependent killing, or you know this one like respiratory burst, and oxygen-independent killing. The oxygen-dependent pathway involves the generation of reactive oxygen species, such as superoxide radicals, hydrogen peroxide. Also, we have net oxidase, nitric oxide synthetase that involves in production of NO. These highly reactive radicals molecules react with proteins, lipids, and other biological molecules to kill the pathogen. Most important for you to know that sometimes macrophages, neutrophils with oxygen-dependent killing can can be harmful to cells that not involved in bacterial infection. And in this case, these products that can kill targetedly bacterial cells also can be targeting by killing our own cells. The oxygen-independent pathway involves the destruction of the pathogen via lysosomal like enzymes such as proteases, phospholipases, nucleases, and lysozymes. These enzymes help to kill pathogens primarily by breaking down their cell membrane. However, this is not so effective like oxygen-dependent killing mechanism, and because of this, sometimes this can help our body to be at war with bacterial cells. So effective phagocytosis requires two components, particle internalization and phagosomal maturation. Although most bacteria are successfully internalized and eliminated by phagocytes, several pathogens have developed survival strategies that interfere with the internalization or maturation processes. Some microorganisms interfere with the ability of phagocytes to engulf them, either by scavenging, inhibition, or even degradation. Opsonic antibodies and complement. Other bacteria have become resistant to one or more of the antimicrobial factors of phagocytes. Some species have developed metabolic pathway to counteract acid accumulation inside phagosomes, or have acquired unequally resistant proteins to withstand with low pH. Yet other microorganisms protect themselves by actively degrading themselves from antimicrobial peptides and proteins produced by phagocytes. All of this, like you see, mechanism of immune evasion leads to us to refers to the term incomplete phagocytosis. Some pathogens can even multiply in phagocytic cells, causing phagocytic cells to die, or spreading phagocytic cells through the lymph or blood to the other part of body, in this case causing widespread infection. Here some classical illustration of dissemination of bacteria Neisseria meningitidis, or Neisseria in this case can be also gonorrhea, only meningitis in bloodstream. Meningococcal bacteria, as you see, these diplococci inside phagocytes, and as you see this, you can be sure that you observe incomplete phagocytosis. So, many microbes have evolved strategies to overcome innate defenses, and this situation, the more specialized way of the immune response is required to eliminate the pathogen. The innate immune response plays a crucial role in the generation of an efficient and effective acquired specific immune response. Also, we have some large parasites, such as helminths, that cannot be physically be phagocyted. So, mast cells, basophils, eosinophils are three additional cell types that play roles in innate and also adaptive immune responses. All three share the common property of having cytoplasmic granules, yeah, filled with various inflammatory and antimicrobial mediators. which are released from the cells upon activation. Another common feature of these cells is their involvement in immune responses that protect against helminths and reactions that cause allergic reaction and allergic diseases. We will introduce and discuss their function in more detail in the next lecture. Also, we have another unique cell in innate immunity. The innate immunity also includes several developmentally related subsets of bone marrow derived cells with lymphoid morphology and effector functions similar to T cells, but lacking T cell antigen receptors. They have another types of receptors. We talk about NK cells, natural killer cells. Natural killer cells have cytotoxic effector functions similar to T cells and, more important, CD8 plus cells, cytotoxic T cells. They circulate in the blood and they are present in various lymphoid tissues. Also, you can see that NK cells also possess cytotoxic granules that contain a battery of serine proteases called granzymes, and as well as pore-forming protein called perforin. Activation of NK cells lead to polarization of granules between nucleus and target within minutes, and extracellular release this contains the granules into space between two cells, followed by target cell's death. NK cells are able to recognize and kill target cells by integrate balance of activating and inhibitory signals based on activation and inhibitory receptors on surface on NK cells. Which allows them to distinguish between healthy cells and target cells, those cells that can be virally infected or transformed. So, very interesting mechanism we can observe in NK cells functions. Firstly, this can be no-killing mechanism. No-killing NK cells have balanced activating and inhibitory signals when recognizing healthy cells. The inhibitory signals are delivered by cells MHA's class 1 in this setting. You see, all our immune cells, all other cells of our organism can be recognized by this complex. In other case, target cells, such as those, like we said, infected, viral infected, or transformed oncogenic transforming cells, often overexpress activating ligands on their surface, which are recognized by activating NK cell receptors. But you can see that inhibitory receptors not activated. So, this process overrides any inhibitory signals, triggering induced self-killing and lysis and killing the target cells by perforin and granzyme mechanism. Let's talk about some conclusion of this. The cellular innate immune system is composed of myeloid cells, including macrophages, monocytes, precursors, mast cells, dendritic cells in this case, some granulocytes like neutrophils, eosinophils, basophils, also natural killer cells, also technically lymphocytes, but they're also part of innate immune system. Cells of innate immune system use conserved pump receptors that very reliably recognize highly conserved features of common pathogens. Innate immune system and innate immune cells act as sentinels of infection. and deal with infections through phagocytosis, the main mechanism against bacterial infection. Or can escalate immune responses through the release of soluble mediators that recruit additional cells to the site of infection. We said that multitude of soluble protein recognition molecules belong to several protein families. Remember about galectins, phyloxins, pentraxins. They also serve to detect consort pumps on microorganisms. Some expression like pentraxin and galectin, this is seroactive protein, acute phase proteins, thicolins, mannose binding lectin, complement system more important. Mechanism of the action include opsonization, complement activation, enhanced phagocytic uptake. So, complement system multi-component triggered enzyme cascade is used to attract phagocytic cells, phagocytic cells to the microbes and engulf them. Complement activation always lead to a membrane attack complex that perforates microorganism. There are three main initiation and three main pathways of activation complement system. Firstly, classical pathway, alternative pathway, and lectin pathway. Classical pathway, so-called because it was discovered first, uses a plasma protein called C1q to detect antibodies bound to the surface of a microbe or other structure. The alternative pathway, which was discovered later, but it's phylogenetically older than classical pathway, is triggered when a complement protein, it's been be called C3 protein, directly recognize microbial surface structures, in this case pumps such as bacterial LPS. The lectin pathway or mannose pathway is triggered by plasma protein called mannose binding lectin MBL, which recognize terminal mannose residues on microbial glycoproteins and glycolipids. The activation of different these pathways lead, as I said, to formate of membrane attack complex. Also, it can trigger phagocytosis by opsonizing antigen and also inflammation by attracting phagocytes and neutrophils inside to infection. The activation of the complement system may be initiated by three distinct pathways, all of which lead to formation of C3 convertase and production of 3,3A, which stimulates inflammation, and 3,3B, opsonization function. 3,3B, most important, all these cascades and these proteins can activate each other. And in this case, 30A distinguish in 30A and 30B. 30B initiate the late steps of complement activation to form a protease called SP5 protea convertase. The next component, CYP5, is activated with the following releasing of C5A, and the residual C5B binds to the surface and assembles the terminal components C6, C9 to activate and formate membrane attack complex with freely permeable to solutes and can lead to osmotic release, that means to target cell death. The activation of complement by either pathways gives rise to 3b, and the generation of a number of factors, like we said, 35a, 33a, 35a, that can aid elimination of foreign bacteria and materials. And a number of the above factors are responsible for the process of acute inflammation. Let's introduce you with these inflammation stages. This is the reaction of the body to injury, such as an invasion by infection agent, exposure to a noxious chemical or physical trauma. The signs of inflammation are redness, heat, swelling, pain, and loss in function. The molecular and cellular events that occur during the inflammatory reaction are the vasodilation, increased vascular permeability, and cellular infiltration. Bacterial infection initiates a series of responses through activation of the alternative complement pathway, producing C3A and C5A, as well as through stimulation the tissue-resident macrophages that detect bacterial derived paths. In this case, the 3B component of complement binds to bacteria, opsonizing the latter for more effective phagocytosis by macrophages and neutrophils. Complement activation can also lead to direct lysis of bacteria through assembly of membrane attack complexes. Activation of macrophages by PAMs and complement compounds induces secretion of mediators, different cytokines release, chemokine release of acute inflammatory response that increase vascular permeability and induce neutrophils to migrate invasions of this neutrophil from the blood into the tissue. The neutrophil arrive first and begin to destroy or remove the offending agent. Mononuclear phagocytes arrive on the scene to finish the removal of the residual debris and stimulate tissue repair. The inflammatory process continues until the conditions responsible for its initiation have been resolved. That means that always these macrophages, mononuclear phagocytes, arise to continue healing and return these resident tissues in a zero activated state. In most circumstances, this occurs fairly rapidly, with an acute inflammatory reaction lasting for a matter of hours or days. So, Isaacs and Linderman discovered in 1957 that cells which have been in contact with inactivated virus particles secrete soluble factors which confess cellular resistance to viral infection. In this case was influenza virus, a phenomenon called interference. In years, it became more and more clear that they called type 1 interference system in our primary defense mechanism against viral infections. In fact, humans with genetic defects in the interferon signaling pathway have a bad prognosis as they die at an early age of virus diseases, which would otherwise pose little problem. So, the interferon system, as we called, who discovered these interferons, are a group of signaling proteins made and released by host cell in response to the presence of virus. But nowadays, interferons are proteins that not only involved in antiviral infection, but also they can be in immunomodulatory and antiproliferative activity. There are three types of interferon. Interferon 1 type, interferon type 2, interferon type 3. A number of molecules have been identified, most important interferons: alpha, interferon beta, interferon gamma. Interferon alpha is produced in the leukocytes and lymphocyte cells infected with a virus, while interferon beta produced by fibroblasts or epithelial cells infected with virus. Interferon gamma, on the other hand, is induced by the stimulation of all sensibilized lymphocytes with antigen or non-sensibilized lymphocytes with mitogens in oncological problems. We will describe the major properties of type 1 interferons and the antiviral action of this primary mechanism against viral infection. Thus, the principal activity of type 1 interferon work in concern to combat viral infection by signaling neighboring uninfected cells to destroy RNA and reduce protein synthesis, or signaling neighboring infecting cells to undergo apoptosis or activated immune cells. Type 1 interferons, interferon alpha, interferon beta, are produced by virus-infected cells, like we said, in response to intracellular toll-like receptor signaling, in other sense of viral RNA. If you remember, we talked about PRR receptors, and some of these receptors would be RIC receptors in cytoplasm and node receptors. Type 1 interferons bind to receptors on neighboring uninfected cells and activate some cascades in these cells. This cascade called JAK-STAT signaling pathway, which induce expression of genes whose products interfere with viral replication. It means this activated EFN genes to producing interferon alpha, interferon beta. Type 1 interferons also bind to receptor on infected cells and induce expression of genes whose products enhance the cell's killing. Interferon initiate, modulate, and enhance the adaptive immune response. The signaling event will culminate in the direct interferon-dependent restriction of virus growing can be delivered in two steps, like in previous slide. Transcriptional induction of interferon signalings, interferon synthesis of protein viruses, degradation of viruses RNA, or antiviral mechanism by inhibition of viral gene expression and virion assembly. This is basic mechanism. Another interesting mechanism in antiviral activity, this is in humoral activity, involve cellular innate system. In this case, NK cells also express a subset of toll-like receptors that are focused towards detecting pumps, such as double-stranded RNA that are typically associated with toll-like receptor 3, toll-like receptor 7, and all of this appear to be functional in NK cells and upon engagement of these receptors. NK cells activated by this and their killing potential is enhanced. Interferon alpha and interferon beta also can be important activators of NK cells. The effects of which can increase the killing activity of such cells by to 100-fold. This is excellent, really excellent example of cooperation between cells of the innate immune system. The cytokines produces by macrophages or the cells upon detection of the pathogen results in activation of other cells, NK cells in the present context that may be better adapted with dealing with infection threat. In case of activated NK cells, can amplify immune responses through production of interferon gamma, for example. Very important cytokines that has been a set of activities distinct from that of interferon alpha and interferon beta. We said that interferon gamma immunomodulate, and macrophages respond to interferon gamma by greatly enhancing their microbicidal activities and also by producing other cytokines such as interleukin 12 that shapes the nature of the ensuring immune response by T cells with the adaptive immune system. Also, it can kill by interferon gamma target cells, and NK cells also in this case can be the line between innate immunity and adaptive immunity, because these activated NK cells produces interferon gamma, and interferon gamma activated dendritic cells and enhances antigen presentation to T cells. So, guys, this is all about our introduction to the immunity, and in this case non-specific immunity, and some resume of our lecture. Protective immunity against microbes is mediated by the early reaction of innate immunity and later responses to adaptive immunity. Innate immune responses are stimulated by molecular structure shaped by group of microbes. This means PAMS molecules. The anatomic organization of the immune system permits the rapid delivery of innate immune cells, including neutrophils, monocytes, to sites of infection, and permits a small number of lymphocytes specific for any antigen to locate and respond effectively to that antigen regardless of where in the body the antigen is induced. The cells that perform the majority of effector function of innate immunity, they are phagocytes against bacterial infection, including neutrophils, macrophages, or mast cells, basophils, eosinophils, if you say about some allergy reaction or Helminth's activity. The principal mechanism of innate immunity to extracellular bacteria, most important, that can be three activation: complement activation, phagocytosis, and the inflammatory. And the last case, if we talk about antiviral activity, the primary mechanism, innate mechanism against antiviral activity would be production of interferon type 1. This includes interferon alpha and interferon beta.